Hi, my name is Malia Moore and I'm a freshman at UC Berkeley. Today I want to share with you a new development in biotechnology that's happening here at Cal, CRISPR-Cas. CRISPR works like super selective DNA scissors that can snip out bits of genes and has the potential to work as a tool to edit gene sequences and treat genetic disorders, though that's just the tip of the iceberg. This all sounds very cool, but how does it work and where does it come from? In the 80s, scientists sequencing the genome of E. coli came across an unusual series of repeating identical segments of DNA with no apparent function, separated by unique spacers. During the century, instances of repeating DNA were found in the genomes of many different species of bacteria and archaea, and dubbed clustered regularly interspaced short palindromic repeats, aka CRISPR, and the nearby genes called CRISPR-associated genes, aka Cas genes. As it turns out, CRISPR-Cas is the system used by microbes primarily as adaptive immunity for targeting predatory viruses. Those spacers are actually short sequences of viral DNA that were captured and placed in the CRISPR locus. So, through cutting open and modifying its own DNA, the host remembers the virus, and that information is copied for all future generations. But how are these viral barcodes used to fight future infection, and why should we care? That's where Jennifer Doudna, a professor at UC Berkeley, comes in, with background in RNA and genetics, asking the fundamental question, how? She found that the viral spacers are transcribed, or copied, to produce segments of CRISPR RNA, each representing a piece of stolen viral DNA. These CRRNA pieces are then picked up by proteins that are encoded by the Cas genes. If a known virus releases DNA into the cell, the Cas protein carrying that corresponding CRRNA is programmed to target and selectively cut the invading sequence to disable it. So in its native prokaryotic cell, a CRISPR-Cas system is responsible for targeting a specific sequence and cutting it. Doudna realized that this highly sophisticated system could be harnessed and used as a tool to selectively cut DNA of interest in any given system with much higher precision and programmability than existing biotech methods. Because CRISPR-Cas is found in so many different microbes, there are a variety of Cas proteins to work with that can be applied for different purposes. Such cutting and pasting abilities have been successfully carried out in the Petri dish, and now research groups on a global level are rushing to learn more about CRISPR-Cas and apply and refine this new gene editing technology to different health problems like genetic disorders, or to agriculture, creating supercrops to combat world hunger, or turning penguins purple, and all because Professor Doudna asked a fundamental question. But as with every major development, this new ability comes with a host of ethical questions, such as, just because we can change something, should we? Thanks for watching. Stay tuned for more from CRISPR-Cas.